A. Host-parasite interaction. A. Host-parasite interaction. Bacteria are consistently associated with the body surfaces of animals. Bacteria are consistently associated with the body surfaces of animals. The many bacterial cells on the surface of a human, including the gastrointestinal tract, then there are human cells that make up the animal. And other microbes that are consistently associated with an animal are called the normal flora, or more properly the indigenous microbiota of the animal. These bacteria have a full range of symbiotic interactions with their animal hosts. Why is it called the normal flora? The term flora is used because the majority of the organisms concerned are bacteria. It has been estimated that humans have approximately 10-13 cells in the body and something like 10-14 bacteria associated with them, the majority in the large bowel. Such as viruses, fungi and protozoa are also regularly found in healthy individuals, but form only a minor component of the total population of resident organisms. The organisms occur in those parts of the body that are exposed to, or communicate with, the external environment, namely the skin, nose and mouth, and intestinal and urinogenital tracts. Internal organs and tissues are normally sterile. The main organisms found in these sites are shown in Figure 1. The normal flora is acquired rapidly during and shortly after birth and changes continuously throughout life. Organisms present at any given time reflect the age, nutrition and environment of the individual. It is therefore difficult to define the normal flora very precisely because it is to a large extent environmentally determined. This is well illustrated by data from NASA astronauts who were rendered relatively bacteriologically sterile by antibiotic treatment before their space flights. It takes weeks after the flight for their flora to repopulate, and the repopulating species were precisely those of their immediate neighbors. The bowel flora of children in developing countries is quite different from that of children in developed countries. In addition, breastfed infants have lactic acid streptococci and lactobacilli in their gastrointestinal tract, whereas bottled children show a much greater variety of organisms. Different regions of the skin support different flora. Dry areas have relatively few resident organisms on the surface, whereas moister areas, axillite, perineum, between the toes, scalp, support much larger populations. Locuus epidermidis is one of the commonest species, making up some 90% of the aerobes and occurring in densities of 103 to 104 per square centimeter. S. aureus may be present in the moister regions. Anerythroids occur below the skin surface in hair follicles, sweat and sebaceous glands, Propionibacterium acnes being a familiar example. Changes in the skin occurring during puberty often lead to increased numbers of this species, which can be associated with acne. A number of fungi, including Candida, occur on the scalp and around the nails. They are infrequent on dry skin, but can cause infection in moist skin folds, intertrigo. Both the nose and mouth can be heavily colonized by bacteria. The majority of bacteria here are anaerobes. Common species colonizing these areas include streptococci, staphylococci, diphtheroids and gram-negative cocci. Some of the aerobic bacteria found in healthy individuals are potentially pathogenic, e.g. Staph aureus, Streptococcus pneumoniae, strep, Pyogenes, Neisseria meningitis. Candida is also a potential pathogen. The mucomembranes of the mouth can have the same microbial density as the large intestine, numbers approaching 10-11 per gram wet weight of tissue. Dental caries is one of the commonest infectious diseases in developed countries. The surfaces of the teeth and the gingival crevices carry large numbers of anaerobic bacteria. Plaque is a film of bacterial cells anchored in a polysaccharide matrix, which the organisms secrete. When teeth are not cleaned regularly, plaque can accumulate rapidly in the activities of certain bacteria, notably Streptococcus mutans, can lead to dental decay. Caries, as acid fermented from carbohydrates, can attack dental enamel. The prevalence of dental decay is linked to diet. The pharynx and trachea carry their own normal flora. The flora of the pharynx and trachea may include both alpha and beta hemolytic streptococci as well as a number of anaerobes, staphylococci, including Staph aureus, Neisseria and diphtheroids. The respiratory tract is normally quite sterile, despite the regular intake of organisms by breathing. However, substantial numbers of clinically normal people may carry the fungus Pneumocystis cerevecii, previously known as Picarini, in their lungs. In the gut, the density of microorganisms increases from the stomach to the large intestine. Stomach contents harbor only transient organisms, the acidic pH providing an effective barrier. However, the gastric mucosa may be colonized by acid-tolerant lactobacilli and streptococci. The upper intestine is only lightly colonized, 104 organisms per gram, but populations increase markedly in the ileum, where streptococci, lactobacilli, enterobacteriaceae and bacteroides may all be present. Bacterial numbers are very high, estimated at 10-11 per gram, in the large bowel, and many species can be found. Figure 2. The vast majority, 95–99%, are anaerobes, bacteroides being especially common and a major component of fecal material. Harmless protozoans can also occur in the intestine, e.g. Entamoeba coli, and these can be considered as part of the normal flora, despite being animals. The urethra is lightly colonized in both sexes, but the vagina supports an extensive flora of bacteria and fungi. The urethra in both sexes is relatively lightly colonized, although staph, epidermidis, strep, phacolis and diphtheroids may be present. In the vagina, the composition of the bacterial and fungal flora undergoes age-related changes. Before puberty, the predominant organisms are staphylococci, streptococci, diphtheroids and Escherichia coli. Subsequently, Lactobacillus eryphilus predominates, its fermentation of glycogen being responsible for the maintenance of an acid pH, which prevents overgrowth by other vaginal organisms. A number of fungi occur, including Candida, which can overgrow to cause the pathogenic condition thrush, if the vaginal pH rises and competing bacteria diminish. The protozoan Trichomonas vaginalis may also be present in healthy individuals. Advantages and disadvantages of the normal flora. Some of the species of the normal flora are positively beneficial to the host. The importance of these species for health is sometimes revealed quite dramatically under stringent antibiotic therapy. This can drastically reduce their numbers to a minimum, and the host may then be overrun by introduced pathogens or by overgrowth of organisms normally present in small numbers. After treatment with clindamycin, overgrowth by Clostridium difficile, which survives treatment, can give rise to antibiotic-associated diarrhea or, more seriously, pseudomembranous colitis. Ways in which the normal flora prevents colonization by potential pathogens include the following. Skin bacteria produce fatty acids, which discourage other species from invading. Gut bacteria release a number of factors with antibacterial activity, bacteriocins, colicins, as well as metabolic waste products that help prevent the establishment of other species. Vaginal lactobacilli maintain an acid environment, which suppresses growth of other organisms. The sheer number of bacteria present in the normal flora of the intestine means that almost all of the available ecologic niches become occupied. These species therefore outcompete others for living space. Gut bacteria also release organic acids, which may have some metabolic value to the host. They also produce B vitamins and vitamin K in amounts that are large enough to be valuable if the diet is deficient. The antigenic stimulation provided by the intestinal flora helps to ensure the normal development of the immune system. What happens when the normal flora is absent? Germ-free animals tend to live longer, presumably because of the complete absence of pathogens, and develop no caries. However, their immune system is less well developed and they are vulnerable to introduced microbial pathogens. At the time of birth, humans are germ-free, but acquire the normal flora during and immediately after birth, with the accompaniment of intense immunologic activity. The disadvantages of the normal flora lie in the potential for spread into previously sterile parts of the body. This may happen when the intestine is perforated or the skin is broken, during extraction of teeth, when burritos. Streptococci may enter the bloodstream when organisms from the perianal skin ascend the urethra and cause urinary tract infection. Members of the normal flora are important causes of hospital-acquired infection. Members of the normal flora are important causes of hospital-acquired infection when patients are exposed to invasive treatments. Patients suffering burns are also at risk. Overgrowth by potentially pathogenic members of the normal flora can occur when the composition of the flora changes, e.g. after antibiotics, or when the local environment changes, e.g. increases in stomach or vaginal pH, the immune system becomes ineffective, e.g. AIDS, clinical immunosuppression. Under these conditions, the potential pathogens take the opportunity to increase their population size or invade tissues, so becoming harmful to the host. Symbiotic associations. All living animals are used as habitats by other organisms. None is exempt from such invasion. Even protozoans have their own flora and fauna. As evolution has produced larger, more complex and better regulated bodies, it has increased the number and variety of habitats for other organisms to colonize. The most complex bodies, those of birds and mammals, including humans, provide the most diverse environments, and are the most heavily colonized. As the normal flora demonstrates, pathogenesis is not the inevitable consequence of host microbe associations. Many factors influence the outcome of a particular association, and organisms may be pathogenic in one situation but harmless in another. To understand the microbiologic basis of infectious disease, host microbe associations that can be pathogenic need to be placed firmly in the context of other symbiotic interspecies associations, such as commensalism or mutualism, where the outcome for the host does not normally involve any damage or disadvantage. All associations in which one species lives in or on the body of another can be grouped under the general term symbiosis, literally living together. Symbiosis has no overtones of benefit or harm and includes a wide diversity of associations. Attempts have been made to categorize types of association very specifically, but these have failed because all associations form part of a continuum. Fig. Point 3. Three broad categories of symbiosis: commensalism, mutualism, and parasitism can be identified on the basis of the relative benefit obtained by each partner. None of these categories of association is restricted to any particular taxonomic group. Indeed, some organisms fit into each category depending upon the circumstances in which they live. Figure 4. Commensalism. In commensalism, one species of organism uses the body of a larger species. At its simplest, the commensal association is one in which one species of organism uses the body of a larger species as its physical environment and may make use of that environment to acquire nutrients. Like all animals, humans support an extensive commensal microbial flora on the skin, in the mouth, and in the alimentary tract. The majority of these microbes are bacteria, and their relationship with the host may be highly specialized, with specific attachment mechanisms and precise environmental requirements. Normally such microbes are harmless, but they can become harmful if their environmental conditions change in some way, e.g. Bacteroides, Escherichia coli, Staphylococcus aureus. Conversely, commensal microbes can benefit the host by preventing colonization by more pathogenic species, e.g. the intestinal flora, by producing metabolites that are used by the host, e.g. the bacteria and protozoa in the ruminant stomach. It follows that the normal definition of commensalism is merely one of convenience, as the association can merge into either mutualism or parasitism. Mutualism. Mutualistic relationships provide reciprocal benefits for the two organisms involved. In parasitism, the symbiotic relationship benefits only the parasite. The terms parasites and parasitism are sometimes thought to apply only to protozoans and worms, but all pathogens are parasites. Parasitism is a one-sided relationship in which the benefits go only to the parasite, the host providing parasites with their physiochemical environment, their food, respiratory and other metabolic needs, and even the signals that regulate their development. Although parasites are thought of as necessarily harmful, this is a view colored by human and veterinary clinical medicine, and by the results of laboratory experimentation. In fact, many parasites establish quite innocuous associations with their natural hosts and are not at all pathogenic under normal circumstances, e.g. when their natural host is in good health. The rabies virus, for example, coexists with many wild mammals but can cause fatal disease in humans. This state of balanced pathogenicity is sometimes explained as the outcome of selective pressures acting upon a relationship over a long period of evolutionary time. It may reflect selection of an increased level of genetically determined resistance in the host population and decreased pathogenicity in the parasite, as has happened with myxomatosis in rabbits. Alternatively, it may be the evolutionary norm, and unbalanced pathogenicity may simply be the consequence of organisms becoming established in unnatural, i.e. new hosts. So, like the other categories of symbiosis, parasitism is impossible to define exclusively except in the context of clear-cut and highly pathogenic organisms. The belief that harmless is a necessary characteristic of a parasite is difficult to sustain in any broader view, and the reasons for this are discussed in more detail below. The characteristics of parasitism. Many different groups of organisms are parasitic, and all animals are parasitized. Parasitism as a way of life has been adopted by many different groups of organisms. Some groups, such as viruses, are exclusively parasitic, but the majority include both parasitic and free-living representatives. Parasites occur in all animals, from the simplest to the most complex, and are an almost inevitable accompaniment of organized animal existence. We can see, then, that parasitism has been an evolutionary success. As a way of life, it must confer very considerable advantages. Parasitism has metabolic, nutritional and reproductive advantages. The most obvious advantage of parasitism is metabolic. The parasite is provided with a variety of metabolic requirements by the host, at no energy cost to itself, so it can devote a large proportion of its own resources to replication or reproduction. This one-sided metabolic relationship shows a broad spectrum of dependence, both within and between the various groups of parasites. Some parasites are totally dependent upon the host, while others are only partly dependent. Viruses are completely dependent upon the host for all their metabolic needs. Viruses are at one extreme of the parasite dependency spectrum. They are obligate parasites, possessing the genetic information required for production of new viruses, but none of the cellular machinery necessary to transcribe or translate this information, to assemble new virus particles or to produce the energy for these processes. The host provides not only the basic building blocks for the production of new viruses, but also the synthetic machinery and the energy required. Figure 5. Retroviruses go one stage further in dependence, inserting their own genetic information into the host's DNA in order to parasitize the transcription process. Viruses therefore represent the ultimate parasitic condition and are qualitatively different from all other parasites in the nature of their relationship with the host. The basis for the fundamental difference between viruses and other parasites is the difference between virus organization and the cellular organization of prokaryotic and eukaryotic parasites. And cellular machinery, and multi-enzyme systems for independent metabolic activity and macromolecular synthesis. The degree of reliance on the host for nutritional requirements varies considerably and follows no consistent pattern between the various groups, nor does it follow that smaller parasites tend to be more dependent. For example, some of the largest parasites, the tapeworms, are wholly reliant upon the host's digestive machinery to provide their nutritional needs. All, of course, receive nutrition from the host, but whereas some use macromolecular material proteins, polysaccharides of host origin and digested using their own enzyme systems, others rely on the host for the process of digestion as well, being able to take up only low molecular weight materials, amino acids, monosaccharides. Nutritional dependence may also include host provision of growth factors that the parasite is unable to synthesize itself. All internal parasites rely upon the host's respiratory and transport systems to provide oxygen, although some respire anaerobically in either a facultative or obligate manner. Parasite development can be controlled by the host. The advantage that parasitism confers in reproductive terms makes it vital to coordinate parasite development with the availability of suitable hosts. Indeed, one of the characteristic features of parasites is that their development may be controlled partly or completely by the host, the parasite having lost the ability to initiate or to regulate its own development. At its simplest, host control is limited to providing the cell surface molecules necessary for parasite attachment and internalization. Many parasites, from viruses to protozoa, rely on the recognition of such molecular signals for their entry into host cells, and this process provides the trigger for their replicative or reproductive cycles. Other parasites, primarily the eukaryotes, require more comprehensive and sophisticated signals, often a complex of signals, to initiate and regulate their entire developmental cycle. The complexity of the signal required for development is one of the factors determining the specificity of the host-parasite relationship. Where the availability of one of the signals entails that parasite development can occur in only one species, host specificity is high. Where many host species are capable of providing the necessary signals for a parasite, specificity is low. Disadvantages of parasitism. The most obvious disadvantage of parasitism arises from the fact that the host controls the development of the parasite. No development is possible without a suitable host, and many parasites will die if no host becomes available. For this reason, several adaptations have evolved to promote prolonged survival in the outside world and so maximize the chances of successful host contact, e.g. virus particles, bacterial spores, protozoan cysts and worm eggs. The prolific replication of parasites is another device to achieve the same end. Nevertheless, where parasites fail to make contact with a host, their powers of survival are ultimately limited. Adaptation to host signals can therefore have a reproductive cost, i.e. the loss of many potential parasites. The evolution of parasitism. As so many organisms are parasitic and every group of animals is subject to invasion by parasites, the development of parasitism as a way of life must have occurred at an early stage in evolution and at frequent intervals thereafter. How this occurred is not fully understood, and it may well have been different in different groups of organisms. In many, parasitism most probably arose as a consequence of accidental contacts between organism and host. Of many such contacts, some would have resulted in prolonged survival, and under favorable nutritional circumstances prolonged survival would have been associated with enhanced replication, giving the organism a selective advantage within the environment. Bacterial parasites evolved through accidental contact. In the case of bacteria, it is easy to see how accidental contact in environments rich in free-living bacteria could lead to successful colonization of the gastrointestinal tract and external orifices. Initially, the organisms concerned would have had to be facultative parasites, capable of life both within or outside host organisms. Many pathogenic bacteria still have this property, e.g. Legionella, Vibrio, but selective pressures would have forced others into obligatory parasitism. Such events are of course speculative, but are supported by the close relationship of enteric bacteria such as E. coli with free-living photosynthetic purple bacteria. Many bacteria and related parasites of humans and other mammals may have originated via the route of accidental contact, but it is clear that others have become adapted to these hosts after initially becoming parasitic in other species. Blood-feeding arthropods provide an example of the most obvious route for this, as their parasites have ready access to the tissues of the animals on which the arthropods feed. Many bacterial parasites have evolved to live inside host cells. Bacteria that became parasitic by accidental contact would have lived outside host cells at first and would not have had the advantages of being intracellular. The evolution of the intracellular habit required further modifications to allow survival within host cells, but could easily have been initiated by passive phagocytic uptake. Subsequent survival of a microbe would depend upon the possession of surface or metabolic properties that prevented digestion and destruction by the host cell. The success of intracellular life can be measured not only by the large number of bacteria that have adopted this habit, but also by the extent to which some organisms have integrated their biology with that of the host cell. The endpoint of such integration is perhaps to be seen in the evolution of the eukaryote mitochondrion, which is widely considered to be the product of symbiotically associated heterotrophic purple bacteria, figure 6. The pathway of virus evolution is uncertain. Clearly, parasitism by bacteria, which are undoubtedly ancient organisms, they can be traced back 3 to 5 billion years in the fossil record, dependent upon the evolution of higher organisms to act as hosts. Whether the same is true of viruses is open to question, and depends upon whether viruses are considered primarily or secondarily simple. If viruses evolved from cellular ancestors by a process of secondary simplification, then parasitism must have evolved long after the evolution of prokaryotes and eukaryotes. If viruses are primitively non-cellular, then it is possible that they became parasitic at a very early stage in the evolution of cellular life, at some point when, because of environmental change, independent existence became impossible. A third alternative is that viruses were never anything other than fragments of the nuclear material of other organisms and have in effect always been parasitic. Modern viruses may, in fact, have arisen by all three pathways. Eukaryote parasites have evolved through accidental contact. The evolution of parasitism by eukaryotes is likely to have arisen much as it may have done in prokaryotes, i.e. through accidental contact and via blood-feeding arthropods. Examples can be found among both protozoan and worm parasites to support this view. There are protozoa such as the free-living amoeba Naegleria, which can opportunistically invade the human body and cause severe and sometimes fatal disease. There are several species of nematode worms that can live either as parasites or as free-living organisms. The evolution of parasitism by eukaryotes is likely to have arisen much as it may have done in prokaryotes, i.e. through accidental contact and via blood-feeding arthropods. Examples can be found among both protozoan and worm parasites to support this view. There are protozoa such as the free-living amoeba Naegleria, which can opportunistically invade the human body and cause severe and sometimes fatal disease. There are several species of nematode worms that can live either as parasites or as free-living organisms, Strongyloides stercoralis being the most important in humans. It is likely that trypanosomes, the protozoans responsible for sleeping sickness, were primarily adapted as parasites of blood-feeding flies and only secondarily became established as parasites of mammals. Parasite adaptations to overcome host inflammatory and immune responses. We can view the evolution of parasitism and the adaptations necessary for life within another animal as being exactly analogous to the adaptations necessary for life within any other specialized habitat. The environment in which parasites live is merely one of the many to which organisms have become adapted in evolution, comparable with life in soil, freshwater, saltwater, decaying material and so on. However, it is always necessary to remember that in one major respect parasitism is quite different from any other specialist mode of life. This difference is that the environment in which a parasite lives, the body of the host, is not passive. On the contrary, it is capable of an active response to the presence of the parasite. The attractiveness of animal bodies as environments for parasites means that hosts are under continual pressures from infection, and these pressures are increased when hosts live close together in unsanitary conditions, in climates that favor the survival of parasite stages in the external world. Pressure of infection has been a major influence in evolution. Pressure of infection has been a major selective influence in evolution, and there is little doubt that it has been largely responsible for the development of the sophisticated inflammatory and immune responses we see in humans and other mammals. In evolutionary terms, all infection has its costs to the host because it diverts valuable resources from the activities of survival and reproduction. There has therefore been pressure to develop means of overcoming infection whether or not it causes disease. Of course, this is not the focus of clinical microbiology, which legitimately places emphasis on the costs of infection in terms of frank disease, but it should be remembered because it explains more fully the nature of the continuing battle between host and parasite, the former attempting to contain or destroy, the latter attempting to evade or suppress and why the emergence of new, and the return of old, infectious diseases are a constant threat. Parasites are faced not only with the problems of surviving within the environment they experience initially, but also of surviving in that environment as it changes in ways that are likely to be harmful to them. The inflammatory and immune responses that follow the establishment of infection are the most important means by which the host can control infections by those organisms able to penetrate its natural barriers and survive within its body. These responses represent formidable obstacles to the continued survival of parasites, forcing them to evolve strategies to cope with harmful changes in their environment. The successful parasite is therefore one that can cope with, or evade, the host's response in one of the ways shown in Figure 7. All of these adaptations are known to exist within different groups of parasites, and they are well documented in the case of some of the major human pathogens. Indeed, they are often the very reason why such organisms are major pathogens. Nevertheless, transmission and survival of many parasites depends upon the existence of particularly susceptible host individuals, e.g. children, to provide a continuing reservoir of infective stages. Changes in parasites create new problems for hosts. From what has been said above, it can be appreciated that there is no such thing as a static host-parasite relationship, and that concepts of unchanging pathogenicity or harmlessness cannot be justified. Each relationship is an arms race, changes in one member being countered by changes in the other. Quite subtle changes in either can completely change the balance of the relationship, towards greater or lesser pathogenicity, for example. Perhaps the most important contemporary illustration of this situation is the dramatic and explosive appearance of HIV infections. This group of viruses was originally restricted to non-human primates, but changes in the virus have permitted extensive infections in humans. Of a different nature, but relevant to the general theme, is the acquisition of drug resistance in bacteria and protozoa, Figure 8. The activity of many antibiotics can be blocked by bacterial enzymes coded for by genes located on cytoplasmic DNA in plasmids. The ability of bacteria to transfer plasmids between individual organisms means that strains or species previously susceptible to an antibiotic can acquire the ability to produce such enzymes and so gain antibiotic resistance directly from resistant organisms. These newly resistant forms are then differentially selected under antibiotic treatment, the susceptible individuals being deleted from the population. Although the underlying genetic and metabolic changes do not by themselves influence pathogenicity, the expression of such changes in the face of intense and selective chemotherapy certainly does so. Host adaptations to overcome changes in parasites. Changes in the host can also alter the balance of a host-parasite relationship. A particularly dramatic example is the intense selection for resistant genotypes in rabbit populations exposed to the myxomatosis virus, which took place concurrently with selection for reduced pathogenicity in the virus itself. There are no exactly equivalent examples in humans, but in evolutionary time there have been major selective influences on populations prompting changes to permit survival in the face of life-threatening infections. A good example is the selective pressure exerted by falciparum malaria, which has been responsible for the persistence of many alleles associated with hemoglobinopathies, e.g. sickle cell hemoglobin. Although these abnormalities are detrimental to a varying degree, they persist because they are or were associated with resistance to malarial infection. Malaria has also changed the frequency of certain HLA antigens in areas where infection was severe. Social and behavioral changes can be as important as genetic changes in altering host-parasite relations. Social and behavioral changes can alter host-parasite relations both positively and negatively, Figure 9. Although many bacterial infections of the intestine have declined in importance with changes in human lifestyle, there are other contemporary microbiologic problems in the developed world whose onset can be traced directly to sociologic, environmental and even medical change, Figure 9. A particularly good example is disease arising from domestication of pets, e.g. toxoplasmosis, because it illustrates that human freedom from some infections arises primarily because of lack of contact with the organisms and not from any innate resistance to the establishment of the infection itself. Diseases arising from contact with infected animals or animal products, zoonotic infections, constitute a constant threat that can be realized by behavioral or environmental changes that alter established patterns of human-animal contact. Host-parasite relationships. If a symbiont either harms or lives at the expense of another organism, the host, it is a parasitic organism, and the relationship is called parasitism. In this relationship, the body of the host can be viewed as a microenvironment that shelters and supports the growth and multiplication of the parasitic organism. The parasitic organism is usually the smaller of the two partners and is metabolically dependent on the host. There are many parasitic agents or organisms among the viruses, bacteria, fungi, plants, and animals. Table 1. By convention, when the word parasite is used without qualification, it refers specifically to a protozoan or helminthic, nematode, trematode, cestode organism. Several types of parasitism are recognized. If an organism lives on the surface of its host, it is an ectoparasite. If it lives internally, it is an endoparasite. The host on or in which a parasitic organism either attains sexual maturity or reproduces is the final host. A host that serves as a temporary but essential environment for some stages of development is an intermediate host. In contrast, a transfer host is not necessary for the completion of the organism's life cycle but is used as a vehicle for reaching a final host. A host infected with a parasitic organism that also can infect humans is called a reservoir host. Because, by definition, parasitic organisms are dependent on their hosts. The symbiotic relationship between the host and parasite is a dynamic one. Figure 1. When a parasite is growing and multiplying within or on a host, the host is said to have an infection. The nature of an infection can vary widely with respect to severity, location, and number of organisms involved. Table 2. An infection may or may not result in overt disease. An infectious disease is any change from a state of health in which part or all of the host body is not capable of carrying on its normal functions due to the presence of an organism or its products. Any organism or agent that produces such a disease is a pathogen. Greek patho, disease, and genon, to produce. Its ability to cause disease is called pathogenicity. A primary frank pathogen is any organism that causes disease in a healthy host by a direct interaction. Conversely, an opportunistic pathogen is an organism that is either normally free-living, or a part of the host's normal microbiota, but which may adopt a pathogenic role under certain circumstances, such as when the immune system is compromised. At times an infectious organism can enter a latent state in which there is no shedding of the organism and no symptoms present within the host. This latency can be either intermittent or quiescent. Intermittent latency is exemplified by the herpes virus that causes cold sores, fever, blisters. After an initial infection, the symptoms subside. However, the virus remains in nerve tissue and can be activated weeks or years later by factors such as stress or sunlight. In a quiescent latency, the organism persists but remains inactive for long periods of time, usually for years. For example, the varicella zoster virus causes chickenpox in children and remains after the disease has subsided. In adulthood, under certain conditions, the same virus may erupt into a disease called shingles. Cold sores, chickenpox, varicella, and shingles, herpes zoster. The outcome of most host-parasite relationships is dependent on three main factors. 1. The number of organisms present in or on the host. 2. The virulence of the organism. And 3. The host's defenses or degree of resistance. Usually the greater the number of organisms within a given host, the greater the likelihood of disease. However, a few organisms can cause disease if they are extremely virulent or if the host's resistance is low. A host's resistance can drop so much that its own microbiota may cause disease. Such a disease is sometimes called an endogenous disease because the agent originally comes from within the host's own body. Endogenous diseases can be a serious problem among hospitalized patients with very low resistance. The term virulence, Latin virulentia, from virus, poison, refers to the degree or intensity of pathogenicity. It is determined by three characteristics of the pathogen: invasiveness, infectivity, and pathogenic potential. Invasiveness is the ability of the organism to spread to adjacent or other tissues. Infectivity is the ability of the organism to establish a focal point of infection. Pathogenic potential refers to the degree that the pathogen causes damage. A major aspect of pathogenic potential is toxigenicity. Toxigenicity is the pathogen's ability to produce toxins, chemical substances that will damage the host and produce disease. Virulence is often measured experimentally by determining the lethal dose 50 LD50 or the infectious dose 50 ID50. These values refer to the dose or number of pathogens that will either kill or infect, respectively, 50% of an experimental group of hosts within a specified period. Figure 2. It should be noted that disease can result from causes other than toxin production. Sometimes a host will trigger exaggerated immunological responses, immunopathology, upon a second exposure or chronic exposure to a microbial antigen. These hypersensitivity reactions damage the host even though the pathogen doesn't produce a toxin. Tuberculosis is a good example of the involvement of hypersensitivity reactions in disease. It should be noted that disease can result from causes other than toxin production. Sometimes a host will trigger exaggerated immunological responses, immunopathology, upon a second exposure or chronic exposure to a microbial antigen. These hypersensitivity reactions damage the host even though the pathogen doesn't produce a toxin. Tuberculosis is a good example of the involvement of hypersensitivity reactions in disease. Some diseases also might be due to autoimmune responses. For instance, a viral or bacterial pathogen may stimulate the immune system to attack host tissues because it carries antigens that resemble those of the host, a phenomenon known as molecular mimicry. Streptococcal infections may cause rheumatic fever in this way. Pathogenesis of viral diseases. The fundamental process of viral infection is the expression of the viral replicative cycle in a host cell. The steps for the infectious process involving viruses are that a virus must 1. Enter a host. 2. Contact and enter susceptible cells. 3. Replicate within the cells. 4. Spread to adjacent cells. 5. Cause cellular injury. 6. Engender a host immune response. 7. Be either cleared from the body of the host, establish a persistent infection, or kill the host. 8. Be shed back into the environment. The determinants of pathogenicity are now discussed in more detail. Entry, contact, and primary replication. The first step in the infectious process is the attachment and entrance of the virus into a susceptible host in the host cells. Entrance may be accomplished through one of the body surfaces: skin, respiratory system, gastrointestinal system, urogenital system, or the conjunctiva of the eye. Other viruses enter the host by needle sticks, blood transfusions and organ transplants, or by insect vectors, organisms that transmit the pathogen from one host to another. Some viruses replicate at the site of entry, cause disease at the same site, e.g., respiratory and gastrointestinal infections, and do not spread throughout the body. Others spread to sites distant from the point of entry and replicate at these sites. For example, the enteroviruses enter through the gastrointestinal tract but produce disease in the central nervous system. Mucous membranes. Viral spread and cell tropism. Mechanisms of viral spread vary, but the most common routes are the bloodstream and lymphatic system. The presence of viruses in the blood is called viremia. In some instances, spread is by way of nerves, e.g., rabies virus, herpes simplex, and varicella-zoster viruses. Viruses exhibit cell, tissue, and organ specificities. These specificities are called tropisms, Greek trope, turning. A tropism by a specific virus usually reflects the presence of specific cell surface receptors on the eukaryotic host cell for that virus. Cell injury and clinical illness. Destruction of the virus-infected cells in the target tissues and alterations in host physiology are responsible for the development of viral disease and clinical illness. Some tissue, e.g., intestinal epithelium, can rapidly regenerate after a viral attack and withstand extensive damage. Other tissues, such as nervous system tissues, are not able to regenerate and may never resume normal functioning after damage has occurred. The potential effects of viruses on individual host cells are the result of a complex series of events. There are 4 generally accepted patterns of a viral infection. 1. In lytic infections, the virus multiplies and kills the host cell immediately and new virions are released. 2. In persistent viral infections, the virus lives in the host cell and releases small numbers of virions over a long period of time. This causes little damage to the host cell. 3. In latent infections, the virus resides in the cell but produces no virions. At some later time, the virus can be activated and a lytic infection occurs. 4. Some viruses can transform the host cell into a cancer cell that becomes the focal point for a tumor. Host immune response. Both humoral and cellular components of the immune response are involved in the control of viral infections. Recovery from infection. The host will either succumb or recover from a viral infection. Recovery mechanisms involve nonspecific defense mechanisms and specific humoral and cellular immunity. The relative importance of each of these factors varies with the virus and the disease. Virus shedding. The last step in the infectious process is shedding of the infectious virus back into the environment. This is necessary to maintain a source of viruses in a population of hosts. Shedding often occurs from the same body surface used for entry. During this period, an infected host is infectious and can spread the virus. In some viral infections, such as a rabies infection, humans are dead-end hosts because virus shedding does not occur. Pathogenesis of bacterial diseases. The steps for infections by pathogenic bacteria include 1. Maintain a reservoir. A reservoir is a place to live before and after causing an infection. 2. Initially be transported to the host. 3. Adhere to, colonize, and or invade the host. 4. Multiply, grow, or complete its life cycle on or in the host or the host cells. 5. Initially evade host defense mechanisms. 6. Possess the ability to damage the host. 7. Leave the host and return to the reservoir or enter a new host. The first 5 factors influence the degree of infectivity and invasiveness. Toxigenicity plays a major role in the 6th. Maintaining a reservoir of the bacterial pathogen. All bacterial pathogens must have at least one reservoir. The most common reservoirs for human pathogens are other humans, animals, and the environment. Since the source and or reservoir of the pathogen is part of the infectious disease cycle. Transport of the bacterial pathogen to the host. An essential feature in the development of an infectious disease is the initial transport of the bacterial pathogen to the host. The most obvious means is direct contact, from host to host coughing, sneezing, body contact. Bacteria also are transmitted indirectly in a variety of ways. Infected hosts shed bacteria into their surroundings. Once in the environment, bacteria can be deposited on various surfaces, from which they can be either resuspended into the air or indirectly transmitted to hosts later. Soil, water, and food are indirect vehicles that harbor and transmit bacteria to hosts. Vectors and fomites, inanimate objects that harbor and transmit pathogens, also are involved in the spread of many bacteria. Attachment and colonization by the bacterial pathogen. After being transmitted to an appropriate host, the bacterial pathogen must be able to adhere to and colonize host cells and tissues. In this context, colonization means the establishment of a site of microbial reproduction on or within a host. It does not necessarily result in tissue invasion or damage. Colonization depends on the ability of the bacteria to compete successfully with the host's normal microbiota for essential nutrients. Specialized structures that allow bacteria to compete for surface attachment sites also are necessary for colonization. Bacterial pathogens and many non-pathogens adhere with a high degree of specificity to particular tissues. Adherence factors called adhesins are one reason for this specificity. Adhesins are specialized molecules or structures on the bacteria's cell surface that bind to complementary receptor sites on the host cell surface. They are one type of virulence factor. Virulence factors are bacterial products or structural components, e.g., capsules and adhesins that contribute to virulence or pathogenicity. Invasion of the bacterial pathogen. Entry into host cells and tissues is a specialized strategy used by many bacterial pathogens for survival and multiplication. Pathogens often actively penetrate the host's mucous membranes and epithelium after attachment to the epithelial surface. This may be accomplished through production of lytic substances that alter the host tissue by 1. Attacking the ground substance and basement membranes of integuments and intestinal linings, 2. Degrading carbohydrate-protein complexes between cells or on the cell surface, the glycocalyx, or 3. Disrupting the cell surface. At times, a bacterial pathogen can penetrate the epithelial surface by passive mechanisms not related to the pathogen itself. Examples include 1. Small breaks, lesions, or ulcers in a mucous membrane that permit initial entry, 2. Wounds, abrasions, or burns on the skin surface, 3. Arthropod vectors that create small wounds while feeding, 4. Tissue damage caused by other organisms, and 5. Existing eukaryotic internalization pathways, e.g., endocytosis and phagocytosis. Once under the mucous membrane, the bacterial pathogen may penetrate to deeper tissues and continue disseminating throughout the body of the host. One way the pathogen accomplishes this is by producing specific products and or enzymes that promote spreading. These products are virulence factors. Bacteria may also enter the small terminal lymphatic capillaries that surround epithelial cells. These capillaries merge into large lymphatic vessels that eventually drain into the circulatory system. Once the circulatory system is reached, the bacteria have access to all organs and systems of the host. Growth and multiplication of the bacterial pathogen. For a bacterial pathogen to be successful in growth and reproduction colonization, it must find an appropriate environment, nutrients, pH, temperature, redox potential within the host. Those areas of the host's body that provide the most favorable conditions will harbor the pathogen and allow it to grow and multiply to produce an infection. Some bacteria invade specific cells in which they grow and multiply. Many of these intercellular pathogens have evolved such elaborate nutrient-gathering mechanisms that they have become totally dependent on the host cells. Finally, some bacteria can actively grow and multiply in the blood plasma. The presence of viable bacteria in the bloodstream is called bacteremia. The presence of bacteria or their toxins in the blood often is termed septicemia, Greek septikos, produced by putrefaction, and hema, blood. Leaving the host. The last determinant of a successful bacterial pathogen is its ability to leave the host and enter either a new host or a reservoir. Unless a successful escape occurs, the disease cycle will be interrupted and the microorganism will not be perpetuated. Most bacteria employ passive escape mechanisms. Passive escape occurs when a pathogen or its progeny leave the host in feces, urine, droplets, saliva, or desquamated cells. The clonal nature of bacterial pathogens. The main mechanism bacteria use to exchange genetic information is the transfer of extrachromosomal genetic elements, plasmids, and phages. Many genes that code for bacterial virulence factors are found on plasmids or phages. These mobile genetic elements can transfer virulence factors between members of the same species or different species by horizontal gene transfer. At times the genetic elements are part of highly mobile DNA transposons, and there is a recombination between the extrachromosomal DNA and the chromosome. When this recombination occurs, the genes coding for virulence may become chromosomal. One important result of the conservation of these chromosomal genes is that the bacteria are clonal. Some bacterial pathogens have only one or a few clonal types existing in the environment. For example, the bacterium that causes typhoid fever, Salmonella typhi, has 2 clonal types, whereas there are over 100 clonal types of Haemophilus influenzae, but only a small number are associated with bacterial pneumonia. Regulation of bacterial virulence factors. Some pathogenic bacteria have adapted to both the free-living state and to an environment within a human host. In the adaptive process, these pathogens have evolved complex signal transduction pathways to regulate the genes necessary for virulence. A virulence factor may be present simply because the bacterium has been infected by a phage. Often environmental factors control the expression of the virulence genes. Common signals include temperature, osmolality, available iron, pH, specific ions, and other nutrient factors. Several examples are now presented. The gene for diphtheria toxin from Corynebacterium diphtheriae, the pathogen that causes diphtheria, is carried on the temperate bacteriophage lambda, and its expression is regulated by iron. The toxin is produced only by strains lysogenized by the phages. Expression of the virulence gene of Bordetella pertussis, the pathogen that causes whooping cough, is enhanced when the bacteria grow at body temperature 37 degrees Celsius and suppressed when grown at a lower temperature. Finally, the virulence factors of Vibrio cholerae, the pathogen that causes cholera, were regulated at various levels by many environmental factors. Expression of the cholera toxin is higher at pH 6 than at pH 8 and higher at 30 than at 37 degrees Celsius. Osmolality and available amino acids are also important. Pathogenicity islands. Many bacteria, Yersinia spp, Pseudomonas aeruginosa, Shigella flexneri, Salmonella typhimurium, and enteropathogenic E. coli, are pathogenic because they have large segments of DNA, called pathogenicity islands, that carry genes responsible for virulence. These pathogenicity islands have been acquired during evolution by horizontal gene transfer. A pathogen may have more than one pathogenicity island. An excellent example of virulence genes carried in a pathogenicity island are those involved in protein secretion. So far, 5 pathways of protein secretion, type cyto V, have been described in gram-negative bacteria. A set of approximately 20 genes encode a pathogenicity mechanism termed the type 3 secretion system that enables gram-negative bacteria to secrete and inject virulence proteins into the cytoplasm of eukaryotic host cells. Unlike other bacterial secretory systems, the type 3 system is triggered specifically by contact with host cells, which helps avoid inappropriate activation of host defenses. Secretion of these virulence proteins into a host cell initiates sophisticated, biochemical crosstalk between the pathogen and the host. The injected proteins resemble eukaryotic factors that signal transduction functions, and they are capable of interfering with eukaryotic signaling pathways. Redirection of cellular signaling transduction may disarm host immune responses or reorganize the cytoskeleton, thus establishing subcellular niches for bacterial colonization and facilitating stealth and interdiction of host defense communication lines. Pathogenicity islands generally increase microbial virulence and are not present in non-pathogenic members of the same genus or species. One specific example is found in E. coli. The enteropathogenic E. coli possesses large DNA fragments, 35 to 170 kilobases in size, that contain several virulence genes absent from commensal E. coli. Some of these genes code for proteins that interact in microfilaments within a host intestinal cell. As a consequence, the host cell surface bulges and develops into a cup-like pedestal to which the bacterium tightly binds. A2. Cell fusion in both normal and abnormal cells. Cell fusion is a process in which 2 or more cells become one by merging their plasma membranes. The ability of a cell to fuse to other cells is referred to as fusogenicity. The progeny of cell fusion are known as hybrids. Perhaps the best-known hybrids are hybridomas, which are made by fusing myeloma cells with lymphocytes to produce monoclonal antibodies. Although cells can be easily fused in the laboratory using readily available chemicals, cell fusion in live organisms appears to be a complex, poorly understood, multi-step process that involves cell-cell recognition, cell adhesion, and membrane fusion. Cell fusion is a part of normal development and tissue homeostasis. Our life begins with fusion of our parents' gametes. A pregnancy depends on normal functioning of the placental barrier, the main part of which is the syncytiotrophoblast, a gigantic syncytium, a cell resulting from fusion of numerous cells whose surface area can reach 10 square meters. As the embryo develops, its muscles are formed by fusion of myoblasts into synchytia through a multi-step process that involves products of multiple genes. In the adult body, the maintenance of the bones is controlled in part by osteoclasts, which are multinuclear cells formed by the fusion of mononuclear progenitors. Macroscopic foreign objects, such as a splinter or an implanted device, are encapsulated by foreign body giant cells, FBGC, that attempt to dissolve the intruder and are thought to be formed by fusion of mononuclear precursors. Langhans cells, a variation of FBGC, are found in tuberculosis patients at the sites of local inflammation. The observations that embryonic stem cells may differentiate into multiple cell types through cell fusion suggested a new role of cell fusion in mammalian development, although alternative explanations for these observations have not been ruled out. Studies in the nematode C. elegans provide evidence that cell fusion can be a major part of development, at least in invertebrates. About 1/3 of the cells that are born in this organism are subsequently fused into 44 synchytia in a reproducible and stereotypic way. Remarkably, even the site of a cell that will fuse is predetermined. In summary, cell fusion of normal somatic cells is a tightly controlled process that is restricted to only a few cell types in humans, and results in terminally differentiated multinuclear cells incapable of proliferation. Intriguingly, tumor cells appear to violate strict rules of cell fusion. Cell fusion in cancer. The idea that cell fusion contributes to cancer progression was introduced almost 100 years ago with a proposal that malignancy is a consequence of hybridization between leukocytes and somatic cells. 60 years later, this idea was expanded by proposing that hybridization of tumor cells with lymphocytes results in metastatic cells, and that cell fusion promotes the phenotypic and genotypic diversity of tumors. Several lines of evidence support at least some of these notions. Many tumor cells are fusogenic. The propensity of various tumor cells to fuse spontaneously in tissue culture has been a known, though unexplained, phenomenon for many decades. In fact, this property of tumor cells was used to make somatic cell hybrids before fusion with polyethylene glycol (PEG) or viruses had been developed. Some tumor cell lines are so fusogenic that they fuse spontaneously more efficiently than in the presence of PEG, a difference explainable by the toxicity of this agent. The ability to fuse is not limited to a particular cell or tumor type and can occur between tumor cells as well as between a tumor and a normal cell. Tumor cell fusion is not limited to tissue culture. Human tumor cells injected into hamsters produce metastasis formed by hybrids of human and hamster cells. In another study, treatment of chimeric mice with a carcinogen resulted in tumors, in which about 1% of cells had macroalleles from both parental strains, an observation explainable by cell fusion. Interestingly, despite these observations, cell fusion is rarely, if at all, considered in contemporary studies that use experimental models of cancer. Because the evidence for cell fusion in human cancers is indirect, it is not as compelling as that for experimental tumors. One observation is that premature chromosome condensation (PCC) is observed in tumor cells. PCC is a typical result of a fusion between cells in different stages of the cell cycle, although PCC can be also induced in mononuclear cells, for example by caffeine. Cell fusion could also explain the origin of multinuclear tumor cells in which nuclei undergo asynchronous DNA synthesis or mitosis, even though alternative explanations of this phenomenon are as plausible. It is unclear whether the scarcity of reports on cell fusion in human tumors is due to rarity of this event, to a difficult It is unclear whether the scarcity of reports on cell fusion in human tumors is due to rarity of this event, to a difficulty to detect it, or to insufficient interest in this subject. The last possibility should not be underestimated. For example, the number of reports on apoptosis that were published during the last 10 years is about 100 times higher than that published in the preceding 20. With all likelihood, this difference is due to a change in the subject's popularity rather than a change in incidence or role of apoptosis in studied organisms. Hybrids can be more malignant than the Hybrids can be more malignant than the parental cells. Importantly, while physiological fusion of normal somatic cells produces non-proliferating differentiated multinuclear cells, fusion of tumor cells results in proliferating hybrids. The survival rate of these hybrids in tissue culture can be as high as 1%. Considering that the rate of cell fusion in experimental tumors was also estimated at 1%, a 1cc tumor of about 109 cells may harbor 105 proliferating hybrid cells. The question is, can any of these cells be more malignant than their parents? In fact, one of the early reports on spontaneous cell fusion came from an observation that cultures containing a mixture of two mouse cell lines were occasionally overgrown by a new cell line, whose karyotype was the sum of the parental karyotypes. This observation indicated that the new cell line is a hybrid, and that hybrids can grow faster than their parents. The subsequent studies indicated that, at least in experimental systems, hybrids can be more drug-resistant and more metastatic than the parental cells, figure 1. Cell fusion can contribute to drug resistance. Cell fusion can consolidate resistance to various drugs by combining genes responsible for resistance to various agents. For example, fusion of tumor cells that were resistant to 5-fluorouracil with tumor cells resistant to methotrexate produced hybrids that were resistant to both drugs. An unexpected observation was that the hybrids were resistant to mephalin, a drug to which both parental lines were sensitive. This observation emphasized the difficulty in predicting the diversity that cell fusion can create. Our own experiments indicated that fusion of drug-sensitive transformed cells to primary cells results in heterokaryons that are resistant to apoptosis. Although this effect lasted only a few days, it is not unreasonable to speculate that even a temporary resistance may allow a small fraction of tumor cells to survive a session of chemotherapy. Cell fusion can promote the ability to metastasize. The ability to metastasize is, arguably, the deadliest property of cancers. A proposed link between cell fusion and metastasis can be described as the wolf in sheep's clothing model. The model suggests that a tumor cell becomes metastatic by fusion to normal cells that travel throughout the body freely, such as lymphocytes or macrophages. Several studies support this idea. One study found that a parental cell line, when injected in a mouse, produced hundreds of colonies in the liver, while a derivative line produced only a few. Surprisingly, when a colony formed by the derivative line was injected into a new host, it produced as many colonies as did the parental cell line. The analysis of histocompatibility markers and karyotypes of the host mice, the injected cells, and the resulting metastasis led to the conclusion that the increase in the metastatic potential was due to a fusion with the host cells. This conclusion was supported by the finding that the high metastatic potential can be restored in the derivative cells by fusing them with mouse bone marrow cells before injection. Other studies discovered that cell fusion can determine the tissues into which tumor cells metastasize. For example, fusion of non-metastatic mouse plasmacytoma or myeloma cells with lymphocytes or splenic dendritic cells not only resulted in metastatic hybrids, but the target tissues of metastasis varied with the type of normal cells used as well. One can only wonder whether implications of this finding are considered in developing cancer vaccines, which involves injecting patients with hybrids between tumor cells and normal dendritic cells. Because tissues that are common sites of metastasis are normally rich in macrophages, the wolf in sheep's clothing model argues that tumor cells gravitate to these sites because, as macrophage hybrids, they acquire macrophage tropism. Another possibility is that tumor cells can acquire the sheep's clothing by fusing to a normal cell of a tissue, thus producing a tumor cell that can grow in a new environment. This mechanism is not unlike that proposed for the stem cell differentiation, where a stem cell differentiates by fusing to a resident cell from a host tissue. Cell fusion can increase tumor cell diversity. Perhaps the main property of cancer cells that makes them malignant is the ability to produce diverse progeny. Indeed, as an oncology textbook states, by the time of initial diagnosis, cancers consist of multiple subpopulations of cells with diverse genetic, biochemical, immunologic, and biologic characteristics. How this diversity emerges and how it is maintained is not clear. The evidence that cell fusion can contribute to tumor heterogeneity came from using cell fusion as a tool for somatic cell genetics, in particular to investigate whether malignancy is a dominant or recessive trait. Because hybrids between a highly and a weakly malignant tumor cell line were highly malignant, the initial conclusion was that malignancy is a dominant trait. However, the subsequent studies found that hybrids between tumor cells and normal cells were not tumorigenic. The controversy was resolved by realizing that hybrids become tumorigenic if they lose certain normal chromosomes, an observation that eventually led to the discovery of tumor suppressor genes. A less appreciated outcome of these studies was the conclusion that chromosomal aberrations are hallmarks of hybrids. These aberrations include chromosome non-disjunction, meiotic recombination, translocations, deletions, insertions, and inversions, a list that is remarkably similar to that observed in tumor cells. Although how exactly cell fusion causes these abnormalities is not clear, the result is hardly surprising considering that cell fusion produces cells with a sum of parental chromosomes and more than 2 centrosomes. The aberrant chromosome segregation associated with cell fusion was proposed to explain aneuploidy as a hallmark of cancer cells. Although aneuploidy is a feature of nearly all of more than 20,000 solid tumors analyzed in human, and it was even proposed to be a cause rather than a consequence of cancer, how and why tumor cells become aneuploid is not clear. The observation that tumor cells are prone to cell fusion, the fact that fusion results in polyploidy cells, and the finding that cell fusion is followed by abnormal chromosome segregation, together argue that if cell fusion is a part of cancer progression, then tumor cells should eventually become aneuploid. However, this explanation of aneuploidy by no means excludes other mechanisms, such as mutations that cause abnormal mitosis. Changes in epigenetic regulation that follow cell fusion are another factor that can contribute to tumor diversity. These changes appear to be unpredictable, and gene expression can be selectively silenced, activated, or unchanged. Considering that the human body has about 200 cell types that are different because of their gene expression pattern, it is easy to imagine how cell fusion can produce monsters. In summary, cell fusion can be an engine of genomic and epigenetic variability that has the potential to make cells with new properties at a rate exceeding that achievable by random mutagenesis. Why are tumor cells fusogenic? There is no definitive answer to this question, which is not surprising considering that the phenomenon lost whatever popularity it had before modern experimental approaches capable of dissecting its mechanism were developed. Several factors were proposed to explain formation of hybrids in tumors: viruses, cholesterol crystals, and the natural fusogenicity of macrophages. The ability of viruses to induce cell fusion is mediated by the viral proteins that mediate entry of enveloped viruses into the cell. These proteins are members of the fusion protein family, which includes both viral and cellular proteins. The fusion proteins are identified by a hydrophobic motif, known as the fusion peptide, which is required for the fusogenicity of these proteins. Perhaps any fusion protein or any other agent that induces cell fusion should be investigated for its carcinogenicity. For example, HIV is known to induce syncretia, which suggests that in addition to its known role in disease, it might be useful to investigate whether this ability contributes to malignancies associated with this virus, such as Kaposi sarcoma. Remarkably, the genes that encode human endogenous retroviruses (HERV) or their individual proteins, including the fusion proteins, comprise at least 8% of the human genome. In fact, syncadin, the fusion protein that is thought to mediate formation of the syncadiotrophoblastin and is specifically expressed in placenta, is the envelope fusion protein of HERV-W. This finding indicates that expression of HERV proteins can determine cell fate, and, incidentally, gives support to the hypothesis that placental mammals evolved because our ancestor had the germline infected with a retrovirus. The sheer abundance of HERV genes in the human genome provides an ample opportunity for their deregulated expression. Indeed, HERV particles were found in both normal and tumor tissues. Production of HERV has been associated with multiple sclerosis and cancer, although whether this association is causative is unclear, in part because HERV particles do not appear to be infectious. Perhaps a causal link between HERVs and these diseases can be found by investigating whether these particles induce cell fusion. Cell death is a predominant outcome of cell fusion, which might explain how expression of HERV can lead to multiple sclerosis, while the ability of some cells to survive cell fusion can contribute to cancer, as we discussed. Overall, at least in experimental systems, cell fusion has been linked to several fundamental features of cancer, even though molecular mechanisms that cause this fusion remain obscure. Although one can argue that this link is a peculiarity of experimental tumors, the hypothesis that cell fusion contributes to cancer in humans seems to be equally plausible. Considering that modern tools of experimental biology have not been applied to studies of cell fusion in tumors, there is an abundance of possibilities that are ready to be explored. Considering that modern tools of experimental biology have not been applied to studies of cell fusion in tumors, there is an abundance of possibilities that are ready to be explored. B1. Cell signaling. 1. Cell-to-cell signaling, hormones and receptors. No cell lives in isolation. In all multicellular organisms, survival depends on an elaborate intercellular communication network that coordinates the growth, differentiation, and metabolism of a multitude of cells in diverse tissues and organs. Cells within small groups often communicate by direct cell-cell contact. We will examine how cells communicate by means of extracellular signaling molecules. These substances are synthesized and released by signaling cells and produce a specific response only in target cells that have receptors for the signaling molecules. An enormous variety of chemicals, including small molecules e.g., amino acid derivatives, acetylcholine peptides, and proteins, are used in this type of cell-to-cell communication. The extracellular products synthesized by signaling cells can diffuse away or be transported in the blood, thus providing a means for cells to communicate over longer distances than is possible by chains of direct cell-cell contacts. The extracellular products synthesized by signaling cells can diffuse away or be transported in the blood, thus providing a means for cells to communicate over longer distances than is possible by chains of direct cell-cell contacts. 2. Overview of extracellular signaling. Communication by extracellular signals usually involves 6 steps. 1. Synthesis, and 2. Release of the signaling molecule by the signaling cell. 3. Transport of the signal to the target cell. 4. Detection of the signal by a specific receptor protein. 5. A change in cellular metabolism, function, or development triggered by the receptor signal complex. And 6. Removal of the signal, which often terminates the cellular response. In many eukaryotic microorganisms, e.g., yeast, slime molds, and protozoans, secreted molecules coordinate the aggregation of free living cells for sexual mating or differentiation under certain environmental conditions. Chemicals released by one organism that can alter the behavior or gene expression of other organisms of the same species are called pheromones. 3. Signaling molecules operate over various distances in animals. In animals, signaling by extracellular, secreted molecules can be classified into 3 types: endocrine, paracrine, or autocrine based on the distance over which the signal acts. In addition, certain membrane-bound proteins on one cell can directly signal an adjacent cell. In endocrine signaling, signaling molecules, called hormones, act on target cells distant from their site of synthesis by cells of endocrine organs. In animals, an endocrine hormone usually is carried by the blood from its site of release to its target. In paracrine signaling, the signaling molecules released by a cell only affect target cells in close proximity to it. The conduction of an electric impulse from one nerve cell to another or from a nerve cell to a muscle cell, inducing or inhibiting muscle contraction, occurs via paracrine signaling. The role of this type of signaling, mediated by neurotransmitters, in transmitting nerve impulses. Many signaling molecules regulating development in multicellular organisms also act at short range. In autocrine signaling, cells respond to substances that they themselves release. Many growth factors act in this fashion, and cultured cells often secrete growth factors that stimulate their own growth and proliferation. This type of signaling is particularly common in tumor cells, many of which overproduce and release growth factors that stimulate inappropriate, unregulated proliferation of themselves as well as adjacent non-tumor cells. This process may lead to formation of tumor mass. Some compounds can act in 2 or even 3 types of cell-to-cell signaling. Certain small amino acid derivatives, such as epinephrine, function both as neurotransmitters, paracrine signaling, and as systemic hormones, endocrine signaling. Some protein hormones, such as epidermal growth factor (EGF), are synthesized as the exoplasmic part of a plasma membrane protein. Membrane-bound EGF can bind to and signal an adjacent cell by direct contact. Cleavage by protease releases secreted EGF, which acts as an endocrine signal on distant cells. 4. The 3 stages of cell signaling are reception, transduction, and response. Our current understanding that how chemical massager act via signal transduction pathways had its origin in pioneering work of Earl W. Sutherland, whose research led to a Nobel Prize in 1971. Sutherland and his colleagues were investigating how the animal hormone epinephrine stimulates the breakdown to polymerization of a storage polysaccharide glycogen with liver cells and skeletal muscle cells. Glycogen to polymerization releases the sugar glucose 1-phosphate, which the cell converts to glucose 6-phosphate. The cell can then use this compound, an early intermediate in glycolysis, for energy production. Alternatively, the compound can be stripped of phosphate and released into the blood as glucose that can fuel cells throughout the body. Thus, one effect of epinephrine, which is secreted from the adrenal gland during times of physical or mental stress, is the mobilization of fuel reserves. Sutherland's research team discovered that epinephrine stimulates glycogen breakdown by somehow activating a cytosolic enzyme, glycogen phosphorylase. However, when epinephrine was added to a test tube mixture containing the phosphorylase and its substrate, glycogen, no depolymerization occurred. Epinephrine could activate glycogen phosphorylase only when the hormone was added to a solution containing intact cells. This result told Sutherland 2 things. First, epinephrine does not interact directly with the enzyme responsible for glycogen breakdown. An intermediate step or series of steps must be occurring inside the cell. Second, the plasma membrane is somehow involved in transmitting the epinephrine signal. Thus, Sutherland's early work suggested that the process going on at the receiving end of a cellular conversation can be dissected into 3 stages: reception, transduction, and response. Figure 1. 1. Reception is the target cell's detection of a signal coming from outside the cell. A chemical signal is detected when it binds to a cellular protein, usually at the cell's surface. 2. The binding of the signal molecule changes the receptor protein in some way, thus initiating the process of transduction. The transduction stage converts the signal to a form that can bring about a specific cellular response. In Sutherland's system, the binding of epinephrine to the outside of a receptor protein in a liver cell's plasma membrane leads via a series of steps to activation of glycogen phosphorylase. Transduction sometimes occurs in a single step but more often requires a sequence of changes in a series of different molecules, a signal transduction pathway. The molecules in the pathway are often called relay molecules. 3. In the 3rd stage of cell signaling, the transduced signal finally triggers a specific cellular response. The response may be almost any imaginable cellular activity such as catalysis by an enzyme, such as glycogen phosphorylase, rearrangement of the cytoskeleton, or activation of specific genes in the nucleus. The cell signaling process helps ensure that crucial activities like these occur in the right cells, at the right time, and in proper coordination with the other cells of the organism. 5. Signal reception and the initiation of transduction. When we speak to someone, others nearby may hear our message, sometimes with unfortunate consequences. However, errors of this kind rarely occur between cells. The signals emitted by alpha yeast cell are heard only by its prospective mates, beta cells. Similarly, although epinephrine encounters many types of cells as it circulates in the blood, only certain target cells detect and react to the hormone. The signal receptor is the identity tag on the target cell. A signal molecule binds to a receptor protein, causing the protein to change shape. A cell targeted by a particular chemical signal has molecules of a receptor protein that recognizes the signal molecule. The signal molecule is complementary in shape to a specific site on the receptor and attaches there, like a key in a lock or like a substrate in the catalytic site of an enzyme. The signal molecule behaves as a ligand, the term for a small molecule that specifically binds to a larger one. Ligand binding generally causes a receptor protein to undergo a change in conformation, that is, to change shape. For many receptors, this shape change directly activates the receptor so that it can interact with another cellular molecule. For other kinds of receptors, as we'll see shortly, the immediate effect of ligand binding is more limited, mainly causing the aggregation of 2 or more receptor molecules. B. Most signal receptors are plasma membrane proteins. Most signal molecules are water-soluble and too large to pass freely through the plasma membrane. But, as Sutherland learned for epinephrine, they still influence cellular activity in major ways. Like yeast-mating factors, most water-soluble signal molecules bind to specific sites on receptor proteins embedded in a cell's plasma membrane. Such a receptor transmits information from the extracellular environment to the inside of the cell by changing shape or aggregating when a specific ligand binds to it. We'll see how membrane receptors work by looking at 3 major types: G protein-linked receptors, tyrosine kinase receptors, and ion channel receptors. G protein-linked receptors. A G protein-linked receptor is a plasma membrane receptor that works with the help of a protein called a G protein, figure 2a. Many different signal molecules use G protein and many other hormones and neurotransmitters. These receptors vary in their binding sites for recognizing signal molecules and for recognizing different G proteins inside the cell. Nevertheless, G protein-linked receptor proteins are all remarkably similar in structure. They each have 7A helices spanning the membrane, as shown in figure 3. Loosely attached to the cytoplasmic side of the membrane, the G protein functions as a switch that is on or off depending on which of two guanine nucleotides is attached, GDP or GTP. When GDP is bound, the G protein is inactive. When GTP is bound, it is active. GTP, or guanosine triphosphate, is similar to ATP. GTP, or guanosine triphosphate, is similar to ATP. When the appropriate chemical signal binds as a ligand to the extracellular site of a G protein-linked receptor, the receptor is activated, changing conformation in such a way that it, in turn, can activate a G protein. The receptor binds a specific and active G protein and causes a GTP to displace the GDP, figure 2b. The activated G protein then binds to another protein, usually an enzyme, and alters its activity. These changes are only temporary, however, for the G protein also functions as a GTPase enzyme and soon hydrolyzes its bound GTP to GDP, figure 2c. Now inactive again, the G protein leaves the enzyme. The GTPase function of the G protein allows the pathway to shut down rapidly when the extracellular signal molecule is no longer present. G protein receptor systems are extremely widespread and diverse in their functions. In addition to the functions already mentioned, they are important in embryonic development, as indicated by genetic studies. For instance, mutant mouse embryos lacking a certain G protein do not develop normal blood vessels and die in utero. Furthermore, G proteins aid involved in sensory reception. In humans, for example, both vision and smell depend on such proteins. Similarities in structure among G proteins and G protein-linked receptors of modern organisms suggest that G proteins and G protein-linked receptors evolved very early, possibly as sensory receptors of ancient microbes. G protein systems are involved in many human diseases, including bacterial infections. The bacteria that cause cholera, pertussis, whooping cough, and botulism, among others, make their victims ill by producing toxins that interfere with G protein function. Although drugs for treating infections and other kinds of diseases have often been discovered by trial and error, pharmacologists now realize that up to 60% of all medicines used today exert their effects by influencing G protein pathways. Tyrosine kinase receptors. Among the chemical signals impinging on cells in an animal's body are growth factors, the local regulators that stimulate cells to grow and reproduce. Cell reproduction involves a variety of activities of different parts of the cell, including protein synthesis in the cytoplasm, chromosome duplication in the nucleus, and the rearrangement of elements of the cytoskeleton. Helping the cell regulate and coordinate these activities is a type of receptor specialized for triggering more than one signal transduction pathway at once. The receptor for a growth factor is often a tyrosine kinase receptor, one of a major class of plasma membrane receptors characterized by having enzymatic activity. Part of the receptor protein on the cytoplasmic side of the membrane functions as an enzyme, called a tyrosine kinase, that catalyzes the transfer of phosphate groups from ATP to the amino acid tyrosine on a substrate protein. Thus tyrosine kinase receptors are membrane receptors that attach phosphates to protein tyrosines. Tyrosine kinase receptors often have the structure shown in rough schematic form in figure 4. Before the signal molecule binds, the receptors exist as individual polypeptides. Notice that each has an extracellular signal binding site, an intracellular tail containing a number of tyrosines, and a single X helix spanning the membrane. The binding of a signal molecule to such a receptor does not cause enough of a conformational change to activate the cytoplasmic site of the protein directly. Instead, the activation occurs in 3 steps. 1. The ligand binding causes 2 receptor polypeptides to aggregate, forming a dimer, a protein consisting of 2 polypeptides. 2. This aggregation activates the tyrosine kinase parts of both polypeptides, each of which then 3 phosphorylates the tyrosines on the tail of the other polypeptide. In summary, the effect of the signal molecule of tyrosine kinase receptor is polypeptide aggregation and phosphorylation of the receptor. The receptor protein is now recognized by specific relay proteins inside the cell. Each such protein binds to a specific phosphorylated tyrosine, undergoing a structural change that activates it. The relay protein may or may not be phosphorylated by the tyrosine kinase. One tyrosine kinase receptor dimer may activate 10 or more different intracellular proteins simultaneously, triggering as many different transduction pathways and particular cellular responses. The ability of a single ligand binding event to trigger so many pathways is a key difference between these receptors and G protein receptors. Abnormal tyrosine kinase receptors that aggregate even without ligand cause some kinds of cancer. Ion channel receptors. Some membrane receptors of chemical signals are ligand-gated ion channels. These channels are protein pores in plasma membrane that open or close in response to chemical signal, allowing or blocking the flow of specific ions such as Na+ or Ca2+. Like the other receptors we have discussed, these channel proteins bind a signal molecule as a ligand at the specific site on their extracellular side, figure 5. The shape change produced in the channel protein immediately leads to a change in the concentration of a particular ion inside the cell. Often this change directly affects cell functioning in some way. At a synapse between nerve cells, for example, it may trigger an electrical signal that propagates down the length of the receiving cell. Ligand-gated ion channels are very important in the nervous system, as are gated ion channels that are controlled by electrical signals. Intracellular receptors. Not all signal receptors are membrane proteins. Some are proteins located in the cytoplasm or nucleus of target cells. To reach such a receptor, a chemical messenger must be able to pass through the target cell's plasma membrane. A number of important signaling molecules can do just that, either because they are small enough to pass between the membrane phospholipids or because they are themselves lipids and therefore soluble in the membrane. Chemical messengers with intracellular receptors include the steroid hormones and thyroid hormones of animals, which are lipids, the small gaseous molecule nitric oxide No, which do not have known receptor and directly activate guanylate cyclase to produce cGMP as secondary messenger, and certain small signal molecules used by bacteria. The behavior of testosterone is representative of steroid hormones, secreted by cells of the testis. The hormone travels through the blood and enters cells all over the body. In target cells, those with testosterone receptor molecule in their cytosol, the hormone binds to the receptor, activating it. The active form of the receptor protein can then bind to and turn on genes in the nucleus that control male sex characteristics. Thus, the receptor carries out the complete transduction of the signal. Prostaglandins, thromboxanes, prostacyclins are nonpolar but have membrane receptors. C. Signal transduction pathways. When signal receptors are plasma membrane proteins, like most of those we have discussed, the transduction stage of cell signaling is usually a multi-step pathway. One benefit of such pathways is signal amplification. If some of the molecules in a pathway transmit the signal to multiple molecules of the next components in the series, the result can be a large number of activated molecules at the end of the pathway. In other words, a very small number of extracellular signal molecules can produce a major cellular response. Moreover, multi-step pathways provide more opportunities for coordination and regulation than simple systems do. D. Pathway relay signals form receptor to cellular responses. The binding of a specific extracellular signal molecule to receptor in the plasma membrane triggers the first step in the chain of molecular interactions, the signal transduction pathway that leads to a particular response within the cell. Like falling dominoes, the signal-activated receptor activates another protein, which activates another molecule and so on until the protein that produces the final cellular response is activated. The molecules that relay a signal from receptor to response, sometimes called relay molecules, are mostly proteins. The interaction of proteins is a major theme of cell signaling. Indeed, protein interaction is a unifying theme of all regulation at the cellular level. Keep in mind that the original signal molecule is not physically passed along a signaling pathway. In most cases, it never even enters the cell. When we say that the signal is relayed along a pathway, we mean that certain information is passed on. At each step the signal is transduced into a different form, commonly a conformational change in a protein. Very often, the conformational change is brought about by phosphorylation. Protein phosphorylation, a common mode of regulation in cells, is a major mechanism of signal transduction. We have already seen how phosphorylation is involved in the activation of tyrosine kinase receptors. In fact, the phosphorylation of proteins is a widespread cellular mechanism for regulating protein activity. The general name for an enzyme that transfers phosphate groups from ATP to a protein is protein kinase. Unlike receptor tyrosine kinases, most cytoplasmic protein kinases act not on themselves, but on other substrate proteins. Also, they phosphorylate their substrates on either of two amino acids, serine or threonine. Such serine-threonine kinases are widely involved in signaling pathways in animals, fungi, and plants. Many of the relay molecules in signal transduction pathways are protein kinases, and they often act on each other. Figure 6 shows a hypothetical pathway containing 3 different protein kinases. This sequence is similar to many known pathways, including those triggered in yeast by mating factors and in animal cells by many growth factors. A cascade of protein phosphorylation, each, transmits a signal bringing with it a conformational change. Each shape change results from the interaction of the charged phosphate groups with charged and polar amino acids. The addition of phosphates often changes a protein from an inactive form to an active form, although in other cases phosphorylation decreases the activity of the importance of protein kinases can hardly be overstated. Fully 1% of our own genes are thought to code for protein kinases. A single cell may have hundreds of different kinds, each with specificity for a different substrate protein. Together they probably regulate a large proportion of thousands of the proteins in a cell. Among these are most of the proteins that, in turn, regulate cell reproduction. Abnormal activity of such a kinase frequently causes abnormal cell growth and contributes to the development of cancer. For a cell to respond normally to an extracellular signal, it must have mechanisms for turning off the signal transduction pathway when the initial signal is no longer present. The effects of protein kinases are rapidly reversed in a cell by protein phosphatases, enzymes that remove phosphate groups from proteins. At any given moment, the activity of a protein regulated by phosphorylation depends on the balance in the cell between active kinase and active phosphatase molecules. When the extracellular signal molecule is not present, active phosphatase molecules predominate, and the signaling pathway and cellular response shut down. Certain small molecules and ions are key components of signaling pathways, second messengers. Not all components of signal transduction pathways are proteins. Many signaling pathways also involve small, non-protein, water-soluble molecules or ions, called second messengers. The extracellular signal molecule that binds to the membrane receptor is a pathway's first messenger. Because second messengers are both small and water-soluble, they can readily spread throughout the cell by diffusion. For example, as we'll see shortly, it is a second messenger called cyclic AMP that carries the signal initiated by epinephrine from the plasma membrane of a liver or muscle cell into the cell's interior, where it brings about glycogen breakdown. Second messengers participate in pathways initiated by both G-protein-linked receptors and tyrosine kinase receptors. The two most widely used second messengers are cyclic AMP and calcium ions, Ca2+. A large variety of relay proteins are sensitive to the cytosolic concentration of one or the other of these second messengers. Cyclic AMP. Once Earl Sutherland had established that epinephrine somehow causes glycogen breakdown without passing through the plasma membrane, the search began for the second messenger. He coined the term that transmits the signal from the plasma membrane to the metabolic machinery in the cytoplasm. Sutherland found that the binding of epinephrine to the plasma membrane of a liver cell elevates the cytoplasmic concentration of a compound called cyclic adenosine monophosphate, abbreviated cyclic AMP or CAMP. An enzyme built into the plasma membrane, adenylyl cyclase, converts ATP to CAMP in response to an extracellular signal, in this case, epinephrine. Adenylyl cyclase becomes active only after epinephrine binds to a specific receptor protein. Thus the first messenger, the hormone, causes a membrane enzyme to synthesize CAMP, which broadcasts the signal to the cytoplasm. The CAMP does not persist for long in the absence of the hormone, because another enzyme converts the CAMP to an inactive product, AMP. Another surge of epinephrine is needed to boost the cytosolic concentration of CAMP again. Subsequent research revealed that epinephrine is only one of many hormones and other signal molecules that trigger pathways involving CAMP. It also brought to light the other components of CAMP pathways, including G proteins, G protein-linked receptors, and protein kinases. Figure 7. The relay molecule immediately after CAMP in a signaling pathway is usually protein kinase A, serine-threonine kinase. Cyclic AMP activates this kinase. The active kinase then phosphorylates various other proteins, depending on the cell. Further fine-tuning of cell metabolism is provided by other G protein systems that inhibit adenylyl cyclase. In these systems, a different signal molecule activates a different receptor, which activates an inhibitory G protein. Now that we know about the role of CAMP in G protein signaling pathways, we can explain in molecular detail how certain microbes cause disease. Consider cholera, a disease that is frequently epidemic in places where the water supply is contaminated with human feces. People acquire the cholera bacterium, Vibrio cholerae, by drinking contaminated water. The bacteria colonize the lining of the small intestine and produce a toxin, which in this case is an enzyme that genetically modifies a G protein involved in regulating salt and water secretion. Because the modified G protein is unable to hydrolyze GTP to GDP, it remains stuck in its active form, continuously stimulating adenylyl cyclase to make CAMP. The resulting high concentration of CAMP causes the intestine cells to secrete large amounts of water and salts into the intestines. An infected person quickly develops profuse diarrhea and if left untreated can easily die from the loss of water and salts. Calcium ions and inositol phosphate. Many signal molecules in animals, including Many signal molecules in animals, including neurotransmitters, growth factors, and some hormones, induce responses in their target cells via signal transduction pathways that increase the cytosolic concentration of calcium ions, Ca2+. Calcium is even more widely used than CAMP as a second messenger. Increasing the cytosolic concentration of Ca2++ causes many responses in animal cells, including muscle cells to contraction, secretion of certain substances, and cell division. In plant cells, calcium functions as a second messenger in signaling pathways. Plants have evolved for coping with environmental stresses, such as drought or cold. Cells use Ca2+ as a second messenger in both G protein pathways and tyrosine kinase receptor pathways. Although cell always contains Ca2+, this ion can function as a second messenger because its concentration in a cytosol is normally much lower than the concentration outside the cell. In fact, the level of Ca2+ in the blood and extracellular fluid of an animal often exceeds that in a cytosol by more than 10,000 times. Calcium ions are actively transported out of cell and are actively imported from the cytosol into the